I'm going to talk about understanding clinical trials for T-cell lymphoma. It's one of the main things that patients are concerned with. What new drugs are out there? What are clinical trials? Should I enroll in a clinical trial? Clinical trials are a kind of research which is designed to evaluate and test new interventions, such as drugs or medications. Clinical trials are often conducted in 4 phases, and I'm going to go through each one of those for you. Phase 1 trials are trials where we're testing a new drug or treatment for the first time. Usually, we do this in a small group of patients. The researchers want to evaluate the safety of the treatment, try to find the appropriate dose, and then identify side effects. In a phase 2 trial, the experimental drug or treatment is given to a larger group of patients to see if it's effective and to further evaluate its safety. In phase 3 trials, the experimental treatment or drug is given to yet a larger group of people. The researchers are attempting to confirm its effectiveness, monitor side effects, compare it to other commonly used treatments, and collect information that will allow the drug to actually be registered by the FDA. And then phase 4 trials are conducted usually after the FDA has already approved the drug, and they're primarily focused on looking at the drug's risks, long-term benefits, and best use. I'm going to give you an example now by showing you 2 real-life clinical trials that are ongoing or planned in our cancer center at Yale. The first one is an example of a phase 1 trial, and this is the actual trial that I'm showing you. This is an open-label, multi-center, non-randomized phase 1 trial, which is a dose escalation trial to evaluate the safety, tolerability, pharmacokinetics, and preliminary efficacy of this drug, ONO-4685, as a monotherapy in patients with relapsed or refractory T-cell lymphoma. So the main goals of this phase 1 study, as stated, are number 1, to determine safety of the drug, number 2, pharmacokinetics, which is how long the drug lasts in the body, and number 3, an efficacy signal, so does it actually work? And again, this drug is being used in T-cell lymphoma. So the next question is, what is the drug? How do you define this new treatment? In this case, the drug is a CD3 bispecific antibody, a novel cancer immunotherapy, which is mainly focused on engaging effector T cells and bringing them in adjacent microenvironment to tumor cells. So hooking onto the T cells, which are going to kill the tumor cell, and then bringing them into the same environment as the tumor cell. So these two can crosstalk, and the T cell can eventually kill the tumor cell. And so basically, this particular drug is targeting human T cells and looking for PD-1 expressing tumor cells. PD-1 is a receptor expressed on certain types of activated T and B cells, but also it's expressed in certain lymphomas such as PTCL and CTCL. The next slide shows you the treatment, so the actual dose escalation treatment protocol with this drug. So when we explore a new drug, we want to, again, look at different doses to determine which doses are safe, and also which doses are effective. So generally, we start at a low dose called cohort 1, and then we escalate as we move along to cohort 2 and cohort 3. Cohorts are just collections of patients. So for instance, there would be 3 patients, up to 6 patients, but no less than 3 in each one of these cohorts. Once one of them is finished and the drug is deemed to be safe, then you'd move to the next one and to the next one. So the point of doing this dose escalation is to determine an optimal dose level that's not toxic. And then once you determine that optimal dose level, then you can do an expansion and treat more patients with specific diseases. In this trial, it would be CTCL patients and PTCL patients. So the dose escalation starts with a very low dose and escalates in groups of 3 to 6. Each cohort is examined for toxicity, and if there's no significant toxicity or side effects, then the next cohort is opened and filled with, again, with another 3 to 6 patients. And the same process keeps going on and on and on until you get to a maximum tolerated dose or a dose that you feel is the best maximum biologically most effective dose. And then you expand, again, looking at specific patient populations. The expansion phase gives you a recommended dose and gives you a signal for where the drug is going to work in a larger phase 2 or phase 3 study. So the next step would be then you would conduct a phase 2 study in 1 or 2 of these populations using the recommended dose, and then move on from there. And again, this just kind of shows you the dosing and scheduling. So one of the things that you explore different doses, but you also might explore different schedules of the drug. So you might start at a lower dose and then increase the dose, for instance. And now this is a very important point in clinical trials because, in fact, the FDA has weighed in on how these drugs are developed. And the FDA, in this new initiative called Project Optimus, is attempting to reform the whole dose escalation scheme that I showed you. Basically, what I showed you is that the doses are stepped up from one group of patients to the next until you get to a maximum dose that has toxicity. Well, the FDA is saying, well, maybe we don't actually need to treat patients at a toxic dose. Maybe we should explore lower doses, and maybe we should explore different schedules as well. And so Project Optimus was just initiated back in 2021, and it's really changed the way we do clinical trials for new drugs. And this slide just kind of shows you the new paradigm now for drug development, especially in orphan diseases like cutaneous T-cell lymphoma, where there are very few patients to enroll in clinical trials. So we're not going to be able to do big trials with thousands of patients looking at different doses in these rare diseases. So we have to really optimize the trial. And so basically, the new schema, again, is a phase 1. It's a dose escalation scheme where you keep treating with higher and higher doses. But what they recommend is that you actually look for the minimally effective dose. So you're looking not only for a maximum dose, but also you're looking for a minimally effective dose. So you determine the minimally effective dose, then you determine the maximum tolerated dose based on perhaps toxicity. And then what you do is instead of going into the phase 2 with this maximum dose, you actually then look at a number of different doses in between minimally effective and maximum tolerated. So then you have a randomization with, in this case, 3 different dose levels. So you're really kind of trying to explore what each one of these doses is going to do and whether, in fact, there is a dose that's better than another dose. Now, I just want to give you an example of how something like this played out recently with one of our drugs for cutaneous T-cell lymphoma. And I'm going to show you a slide later on about the ONTAC drug, which is now called E7777. When we actually did clinical trials with that drug, what we learned is that we used a dose of 9 and 18 mics per kilogram. So we had 2 different dose levels in the trial. And what we learned is that there was no difference between those 2 doses with respect to safety or efficacy. So actually both doses worked. The low dose worked as well as the higher dose. Now, there were some minor differences in terms of whether one worked better for more advanced patients, but nevertheless, the FDA actually approved that drug, not at 1 dose, but at 2 doses. And I think we may start seeing more of that as we move into this new era in drug development, where we're looking more closely at what actual doses are going to be effective for patients. Okay, so now moving on to an actual clinical trial. So we've determined how we're going to do the trial, phase 1, 2, or 3. And next we have the inclusion criteria. The inclusion criteria define the patients that are eligible for the trial. So if you're being screened for a clinical trial, basically what you're told is that you need to meet certain exclusion and inclusion criteria. So you have to have the disease that the trial is focusing on. You may have to have a specific stage of the disease. in many of these trials, you have to be relapsed. So many of these new therapies are looking at patients that have already failed at least one therapy. In this case, you have to have failed two therapies. You have to have measurable disease on a PET scan or something we can measure, like skin lesions. And then you have to be in fairly good shape with respect to your liver, your kidneys, and your other organs so that you don't have any damage to those organs on the trial. And then there are exclusion criteria. So for instance, some of these therapies may not work in certain diseases. So in this case, adult T-cell leukemia is excluded. In addition, there may be some other prior cancer therapies that would make you not likely to have a good response to this new treatment if you'd had those. In this case, it talks about drugs that are being given within 4 to 6 weeks of the time you get the experimental drug. Many of the trials exclude people that have already had transplant. And I think this is a real disadvantage for us because our patients who failed the transplant are not able to get these new drugs. And then there are other criteria as well. So the investigator is going to look at these inclusion and exclusion criteria to determine whether you're eligible for the trial and also just to determine whether they think that the trial would be safe for you. Another aspect of the trial is what we call the schedule of assessments. And again, this is something that directly involves you as a patient because this is really what you're going to have to do if you're on the trial. And so the trials all define this in great detail. They define whether or not you're an inpatient. In this case, there is a hospitalization for part of this clinical trial. It tells you when you're going to get the drug, so what days you're going to get the drug over the course of the treatment period. It also tells you what tests are going to be done, so what lab tests are going to be done on a regular basis. And it pretty much spells out all of the details that you need to know for the trial. Also, I'll just mention that when the Food and Drug Administration looks at these trials, they look at this with a fine-tooth comb. And so they really dig deep into our records to make sure that, in fact, we did all of the things that we said we were going to do. So now I'm going to show you a phase 3 clinical trial. Again, this is a real world trial. This is a trial that we're opening at Yale that will enroll patients. Because it's a phase 3 trial, it's also open in many other treatment centers in the United States as well. A phase 3 trial, again, is oftentimes a registrational trial that's designed to meet FDA guidelines for a drug approval. So in this case, it's a drug or a treatment that's already been tried in phase 1 studies and then has moved to phase 2 studies showing that it's effective. The phase 3 trial then is hoping to prove its efficacy or its effectiveness when compared to other treatments available for that particular disease so that the FDA can then go ahead and approve the drug. Oftentimes, the phase 3 trials are randomized. So in this case, you have a patient population of T-cell lymphoma patients. In this case, the study is hoping to enroll 150 patients. Patients, when they're enrolled, are randomized. The randomization is done by the computer into either an arm that gets the active treatment or a standard of care arm. And standard of care would be drugs that we use anyway, such as, in this case, belinostat or pralatrexate. And then based on this randomization, you would go into one arm or the other arm, and you would get your treatment. And then at a certain point, which is usually 2 or 3 months later, we would determine whether or not you're responding. So if you randomize to the standard of care and you don't get the new drug, then in this particular trial, you could actually cross over if you're not responding. So when you get to that 2 to 3 month period where we do a PET scan or assess your skin and see whether you respond, if you're not responding in the standard of care, you're able to then go into the treatment arm. And this is a very important aspect when you look at these randomized trials. Are you going to have an opportunity to actually get the experimental treatment if you randomize to the control arm? So as I mentioned, these are randomized trials that oftentimes enroll a large number of patients. For instance, the clinical trial that led to the approval of mogamulizumab in CTCL patients enrolled over 400 patients. So there were over 200 patients in the active treatment arm and over 200 patients in the control arm. Now, again, I mentioned the crossover. So not all trials have a crossover. So in some trials, you're not going to be able to get the experimental arm if you randomize to the standard of care. And as you can imagine, that sometimes affects the decision-making on the part of a patient as to whether they want to enter that clinical trial. Well, these phase 3 trials usually take years to complete. So these are usually 5, maybe 7 years to actually get through enrolling all the patients, allowing them to be treated, and then following them. And these all have predefined endpoints agreed upon with the FDA. So the sponsor has to tell the FDA in advance, we're doing this trial. We expect that the control arm, that the experimental arm will have a 20% overall benefit in terms of progression-free survival or whatever their endpoint is. So they have to define that, and they have to say these are our parameters to determine whether our new treatment is really worthy of being approved. And so oftentimes, of course, the companies will meet those endpoints in their clinical trials, and then they go on to go to the FDA and the drug gets approved. So in this particular trial, the study period is up to 2 years of treatment and 2 years of follow-up. So basically, we're talking about 4 years for each patient from the time that they're enrolled. And again, this is going to open at a large number of treatment sites at 25 treatment centers around the United States. And here's another point with these clinical trials. This trial is hoping to enroll 150 patients that can be randomized. In order to do that, they have to actually screen over 170 patients because there are going to be patients who are screened, and based on those inclusion and exclusion criteria, they may not actually make it onto the trial. So oftentimes these large trials screen a significant number of patients in order to make their endpoints. So if there is a phase 3 clinical trial open to you, I would strongly recommend that you consider it. It's very important for us to actually get patients on these trials and to accrue them as quickly as possible so that we can get these new treatments to you, directly to you in the clinic. Okay, so, and I just want to say one thing now about how we actually develop new drugs and where we get these new drugs from. So obviously, a new drug is a drug that's going to target a specific aspect of the cancer or the tumor microenvironment. And these new drugs oftentimes come because of discoveries in the biology of the disease. So as our basic researchers are examining tissues from patients and tumor cells and looking at different animal models of the disease, they're able to identify specific aberrations or abnormalities in the tumor cells. So for instance, there might be certain receptors that are mutated. There might be certain pathways within the cell that involve multiple genes that are mutated. There might be certain mutations in the DNA and different genes and different proteins that are affecting the DNA of the cell. There might be others that are on the surface of the cell, other proteins on the surface of the cell that are mutated. So all of these things are, of course, studied extensively in the laboratory. And if, for instance, we identify a mutation or a pathway that is somehow abnormal in the cancer cell, we can develop a treatment specifically toward that pathway. So, for instance, the JAK-STAT pathway is a surface receptor. It's a signal transduction molecule that stimulates the proliferation and is important in many of our T-cell lymphomas. And so now we actually have drugs that target this, specifically bind to this and inhibit this pathway. We also have many drugs that target epigenetic abnormalities affecting the way the DNA is wound into chromosomes. And again, these drugs target specific proteins that we know are implicated in this particular process. So really finding a new drug goes back to the biology, which is finding a new target. And that's why it's critically important to actually fund basic research into these diseases. I'm going to just say a little bit now about immunotherapy. Again, the two examples that I mentioned to you of these new therapies are focused on immunomodulation or immunotherapy of disease. I know that you heard from Tarshin, one of our prior speakers in the program, about some of the newer drugs that we're using and some of our current therapies. But where do we go from here? So what is the next generation of treatment going to look like? And many of, I think, the next generation drugs are going to be immunotherapies rather than standard chemotherapies. So if we look at the whole field of immunotherapy, there are a number of different ways that we can use immunotherapy to target a cancer cell. So in this case, I'm showing you a B cell because B cell lymphomas currently have many more of these immunotherapies than T cells do. So I wish and I hope that a couple of years from now, this diagram is going to be a T cell lymphoma, and we're going to have all these different therapies for our, specifically for our T cell patients. But in the case of B cell lymphoma, you can see that there are different kinds of cell-based therapies, antibody-based therapies, and toxin therapies. So if you focus then on T cells, for the T cell lymphomas, we certainly have monoclonal antibodies, which are directed against specific proteins on the tumor cell. An example of that would be mogamulizumab. We also have monoclonal antibodies that are hooked up to toxins, that are hooked up to toxic drug moieties. And an example of that is brentuximab vedotin, and also denileukin diftitox, which is ONTAC. We have what we call dual antigen retargeting proteins. And so these are antibodies that basically have two different epitopes that recognize two different cell types. So in this case, you can see these B cell directed, what we call bispecific antibodies. First of all, they engage the T cell, the activated T cell through CD3. And then on the other arm, they engage the B cell through a receptor called CD20. So in the case of our T cell lymphomas, we have bispecific antibodies that target CD30 on the tumor cell, and then CD16A on the effector immune cell. And also we have one that targets CD3 on the T cell, and then PD1, which is expressed on the tumor cells on some of our T cell lymphomas. And others are being developed as well. So these bispecific antibody engagers, I think, are going to be very useful in the future. And there are 3 of these that are FDA approved now for B cell lymphomas. Immune checkpoint inhibitors are also important, and these basically target the part of the immune system that's preventing tumor cells from being killed. And pembrolizumab is one of the antibodies that we use that's an immune checkpoint inhibitor. There are others as well. And then finally, I'm going to talk about chimeric antigen receptor T cells, which are T cells that are actually engineered to specifically attack and kill tumor cells. So I just want to say one thing about E7777, which is the new derivative of ONTAC. So ONTAC was really the first biologic therapy that was FDA approved for T cell lymphoma, and it was used extensively for CTCL until it was taken off the market by the FDA, primarily because the molecule needed to be cleaned up and put into a different solution to make it a better and more pure drug. So the better and more pure derivative is called E7777. So basically what this is the diphtheria toxin, which is a toxin that causes diphtheria, hooked up to the interleukin-2 receptor, which targets it to T cells. So normal T cells, as well as T cells from patients with cutaneous lymphoma and other types of T cell lymphoma, express the interleukin-2 receptor on the surface. So the denileukin diftitox E777 basically targets the interleukin-2 receptor. It binds to it, and it delivers this toxin, which then goes into the cell and kills the cell. So it kills the cell directly by binding to this receptor. And so when we actually use this drug, and I mentioned This is the one where there were two different doses, 9 and 18 mics per kilogram per day for 5 days of dosing. Basically, this is capable of killing the cells, and the response rate is about 36%. There was a trial that was done in both PTCL and CTCL, and there were many patients who had good responses, as you can see by, these are all people that responded to the drug on that trial. And then subsequently, a CTCL trial was just completed, and this molecule is now at the FDA waiting for a final approval so that it can be given to patients again. And I think that will happen probably by November or December of this year. And just to mention, side effects of this include elevation of your liver functions, lowering of your albumin, and some decrease in blood counts. But again, these are all reversible, very quickly reversible, and patients are able to actually tolerate and stay on the drug. So I'm very excited to see that that drug will come back. Okay, so let's talk about CAR T-cell. CAR T-cell is the newest thing. You may have read about this in the New York Times or on other news media, because it's really proven to be a highly effective way of curing patients who have otherwise incurable cancer. And what it basically does is it harnesses the immune system to do a job. Basically, in the context of having a tumor or a cancer, your normal T cells are supposed to be able to recognize those tumor cells as being aberrant or foreign or expressing aberrant proteins. And your normal immune system is supposed to kill the tumor cells. This probably happens all the time in our bodies, because there are always cells that are mutated in our bodies at all times. And what happens is the immune system clears these cells out before they can become tumors. However, in certain cases, it's unable to do that job, and these cells are allowed to divide and become tumors. So basically, in all of you who have lymphoma, have T-cell lymphoma or other kinds of cancers, you basically have T cells that aren't doing the job. So how do you then make these T cells do the job? So what you do is you turn them into targeted killer cells. So you want to get these T cells activated so they'll go after those tumor cells and kill them. So basically what happens here is that the T cells, and I'll show you a picture in a minute to explain this, but the T cells are taken out of the body and they're injected with a gene that basically activates the T cells and targets them toward the tumor cells. So gives them a target on the tumor cells to go after, and then makes them active so that they will become killer cells. So what happens with CAR T-cell is that you go to the blood bank and you put on one of the machines like you're donating blood. And what happens is that the machine takes the white blood cells, which are mostly T cells, out of your body, puts them into a bag. Those T cells in that bag then undergo a process. So they're sent off to the company that's going to generate the CAR T cell for us. So they are then genetically modified with a viral vector, basically, that will give them the coding targeting receptor that they need to target the tumor cells. And also will inject into these T cells the activation genes that will make them into killer cells. So the cells are basically modified with this gene, and then they're expanded in culture to make, a lot of them. And these expanded cells then are put into a bag, and then they come back into the patient. They come back to us, and then we infuse them into the patient. And so basically what happens is cells come out. There's about a 2 to 3 week time frame here from the time we take the cells out till the time we give the cells back. And in that period of time, your cells go to the company. They get genetically modified, they get expanded, they get purified, and they get put back on the airplane to come back to us. So all of that works very well. But the main problem with CAR T-cell is that there's this waiting period to get your cells back. And the other thing is that you don't actually know whether all the cells you're getting back are good cells, and maybe some of them are dying, and maybe something happened during the transportation, and maybe the gene didn't go in very efficiently. So obviously there's a lot of testing that's done to try to make sure that that's optimized. But a better way of doing it would be, obviously, if you could give people off-the-shelf T-cell products. So in other words, what if you didn't use your own cells? What if we were able to take T-cells from normal donors, basically do all this in the lab and put all these on the shelf and basically freeze them all away, so that if a patient comes in and they need a CAR T-cell, you would just take it out of the freezer and give it to the patient. Well, that is exactly what the CRISPR company did. So the CRISPR company, what they did is that they genetically modified normal T cells from normal donors, so that basically they took away all of the recognition signals that would make your T cells be rejected by somebody else's body. So they took away what we call the MHC locus. And then they put in the gene to target those T cells, in this case to CD70 on the tumor cells, and then they activated them. So they basically created what we call off-the-shelf allogeneic CAR T cell product. So this is a trial that we had open at Yale, and this trial was of interest for this audience because it was specifically for patients with cutaneous T-cell lymphoma. And so we treated a number of patients. We were one of 7 or 8 sites in the country. And we showed that 40% of patients had a complete response to this therapy. And in CTCL patients, they responded in the lymph nodes, the skin, and the blood. So, some of our treatments for CTCL are better for blood, say, than they are for skin. Some of them, such as radiation and topical treatments, are better for skin, but not good for blood. But in the case of this therapy, it actually worked across all the different compartments of disease. So looked like a very good therapy for people with CTCL. And I'm just showing you this to give you an idea of how dramatic this cell-based therapy can be. So again, this is a patient with advanced transformed mycosis fungoides, had 5 prior therapies, and in fact was refractory to the last therapy, which was brentuximab. So these CAR T cells target CD70 on the tumor cells. So of course we want to measure that and make sure that the tumor cells express CD70, which of course they did in this gentleman. The patient received the cells, and then 28 days later, after a single infusion of these cells, the patient had a complete response. And so this is a PET scan showing lots of different tumors on the skin. You can see them. And this is a PET scan on day 28, where you can see that just about all of the disease is gone. Very, very effective. And this patient actually has continued to be in complete response. And the side effects of this are really just lowering of some of the blood counts, minimal side effects, and again, very well tolerated by patients. So I think this is really our best example of how an immunotherapy, a cell-based therapy, a specific targeted cell-based therapy can work and can be very effective even with 1 dose. And so we're hoping in our future in CTCL to see the development of more of these kinds of therapies for our patients. And my last slide again just tells you like what is the state of the art now for these new therapies in CTCL. So this is the one I just showed you, the CD70 allogeneic CAR T-cell therapy. Overall, if you looked at CTCL and PTCL, we had a significant number of patients responding. And again, our safety profile was pretty good with not many patients having really bad side effects. There's also another CAR T-cell that's directed against CD5, which is different than CD70. Again, this is an autologous CAR, which means that it's your own cells that are engineered. This one is still experimental. Again, you can see people responded, but interestingly, there were no cutaneous T-cell lymphoma patients that responded. Only 2 were treated. Perhaps if we treat more, we might see some different data here. And then the last thing I want to mention is a bispecific antibody. And if you remember my talk about bispecific antibodies, I told you that it's an antibody that has 2 different arms. One arm is targeting the tumor cell, so that would be CD30. The other arm is targeting a a T cell or an NK cell or one of the effector killer cells that's going to kill that tumor cell. So this treatment is called AMF13. And this is an ongoing phase 2 clinical trials. And so hopefully we'll see some data from that